Hello, my name is Ariel Walter, and today I'm going to be talking with you about regenerative medicine, the future remedy of the human condition. So, humans have often had this desire to live longer. We see this through the Fountain of Youth, Bionic Man. We see that people are often looking for ways to live longer, healthier lives. So what we're going to be looking at today is a field that incorporates molecular biology, genetics, and also engineering into a comprehensive unit to where you can make anything from a diagnostic 3D cell culture, a possible organ on a chip type structure, or even an organ, a functioning organ within the human body. So our main methods are stem cell therapy, tissue engineering, and diagnostic and personalized therapies, which we'll be talking about today. So our first method we're going to be talking about is stem cell therapy. And stem cell therapy is pretty much the use of stem cells, which are regenerative cells that are found within the body, and they can be used in rebuilding tissues or treating disease. And pretty much, we can have two ways of using stem cells. We can either use a patient's own stem cells, or we can use stem cells from another patient. So when you use a patient's own stem cells, there's a lot of benefits and advantages. First of all, the biggest one is probably not having to use immunosuppressant drugs. This is really important when you're looking at disease models, because a lot of diseases can enter the body when the immune system is weakened. And in order for a person or a patient to go ahead and have somebody else's cells or transplants put into them, they have to have immunosuppressant drugs so they won't react to that transplant. So by using a patient's own stem cells, we're able to go ahead and regenerate the body in a way that, say, a transplant would, or stem cell therapy would, but we don't have to use the immunosuppressant drugs. So that just makes the immune system that much better. One of the best things about stem cell therapy is it actually only takes about a month to grow enough cells in order to treat the patient. So we have current research, which is currently autoimmune disease disorders, central nervous system disorders, cancer therapy, chronic wounds, and you can even differentiate stem cells such as adipose stem cells into neuron-like cells, which researchers hope will allow treatment in brain damaged patients. Now we're going to move on to tissue engineering. Tissue engineering actually integrates stem cell therapy into biomaterials engineering. So first of all, biomaterials engineering is the creation of a scaffold. Sometimes a scaffold is created from polymeric materials in the lab. Also, a scaffold can be utilized from human tissue, such as an esophagus, which can have the cells scraped off and therefore be used for a new patient without any of the cellular interactions that cause an immune response. So once we have the scaffold, we can actually seed our stem cells onto the scaffold, and these stem cells can colonize the scaffold. We can then put it into the human body. Say we're working on a bone. Okay, so we have a polymeric scaffold, and we seed it with stem cells. Well, now, over time, those stem cells will actually start to make the bone just like the scaffold did. However, the scaffold will break down, and that will allow it to become an integrated part into the human body that's able to interact, create bone marrow type stem cells, all of those processes in the body. So it actually becomes integrated. It's not just like a metal unit that you put in the body. Finally, really quick, I'm going to go ahead and I'm going to talk about diagnostic and personalized therapy. You have spheroids and organoids. What is a spheroid and an organoid? It's a 3D cell culture. Pretty much the difference is a spheroid is a 3D cell culture of cancerous type cells, and an organoid is a 3D cell culture of healthy type cells. So spheroids can be used when testing out new cancer therapies. Organoids can be used instead for growing, say, either one type of tissue or multiple cell types in one tissue, which most of our organs are composed out of. So say we wanted to try and treat kidney failure, we could actually grow a kidney and try out different treatment options that may be available for kidney failure. Proteomics utilizes protein output to analyze what is happening in the body, so it can actually show the current state of health. This is especially important when trying to analyze what medications might interact with a human body, one patient versus another patient. So it's a lot more of a personalized medicine. Finally, we have organ on a chip, which can be used to make organ systems. It can be utilized with organoids, and it can basically represent the human body system. So thank you for your interest in regenerative medicine, and hopefully in the next coming decades we can see more on this amazing therapy. Thank you for your time.